So thank you for the opportunity to speak to you today about dengue virus and host responses to dengue virus. My name is Ashley St. John. I'm Associate Professor in the Program in Emerging Infectious Diseases at Duke and U.S. Medical School in Singapore. So we know that dengue is caused by dengue virus. It's one of the most prevalent pathogens in the world borne by mosquitoes, and in fact, the most prevalent viral pathogen. And in fact, most people who experience dengue may not even know that they have had a dengue infection. But in rare cases, complications can develop, and usually these involve vascular complications. So you can see here on the right side some of the signs and symptoms of severe dengue, including vascular leakage and hemorrhaging. So up in the left-hand corner, you can see some images from a publication with an autopsy where they looked at various organs after death due to dengue. And you can see that there's a good bit of hemorrhaging in the panel A. They're showing the gut. In panel B, you can see the kidney and that there's quite a bit of hemorrhaging there inside the kidney. In panel C, this is the liver with what looks almost like bruising on the surface, but this is due to the microvascular permeability and hemorrhaging. And in panel D, it's really quite severe. These are the lungs. So this is worst case scenario with dengue, but there's quite a spectrum of disease and many immunological complications, including rash and fever. So we know that a small portion of people will develop severe disease, but usually we think of those processes that result in severe disease as beginning earlier and being immune mediated. So you can see in these time courses of infection what happens to an individual when they're experiencing dengue. So after the mosquito bite, there's a period of time that's an incubation period before the onset of fever. But during this time, the immune response is responding to dengue, detecting it, and beginning to initiate the adaptive immune response, which can come up within a week to 2 weeks following infection. So importantly, there are 4 different serotypes of dengue. So that means that you can experience 4 different types of infection in a lifetime, theoretically. And usually, immunity to one dengue infection can be protective against the same serotype of dengue, but is not protective and may even be associated with more severe disease during a secondary dengue infection. So you can see here on the bottom how these curves are shifted. Although viremia may be a shorter period of time, there is a heightened risk of shock and hemorrhage in those individuals who are experiencing a secondary dengue infection. And importantly, this risk of shock and hemorrhage usually occurs towards the end of the disease time course as viremia is subsiding and as the fever is subsiding. So, my lab is quite interested in these early time points, what happens immediately following the mosquito bite and how that can set up for this downstream severe disease. So when a mosquito bites and injects dengue into the skin, it's not unopposed. There's a good bit of the immune system that is there, ready and waiting to respond. And so you can see here in a whole-mounted tissue of skin how the blood vessels are in blue, and they form a very dense network. And this is what the mosquito is probing and trying to find. But in close proximity to blood vessels are immune cells. And this image shows one type of immune cell, which is called the mast cell. And mast cells are distributed lining blood vessels throughout the connective tissues in the body, very, very densely in the skin, about the same density as dendritic cells and macrophages, which are discussed perhaps a little more often. When we talk about mast cells, we usually talk about their role in allergic responses, how they detect these innocuous environmental antigens, such as shown at the bottom, and result in the diseases that we call asthma, allergy, anaphylaxis, or contact hypersensitivity, which are all thought to be mast cell mediated. And usually in those contexts, mast cells might not respond to these antigens alone, but they do so through interaction with a unique subclass of antibodies called IgE. And this induces cross-linking of the Fc receptors that are shown here on the surface of the mast cell and can trigger mast cell degranulation. So this is, at the top, an image of mast cell degranulation by scanning electron microscopy. So you can see on the left in panel a resting mast cell where they have all of these little granules that are right under the surface. They're ready to go. So this mast cell is armed. And these granules contain inflammatory mediators, not just cytokines and chemokines, which are also there, but in addition to that, many proteases and anticoagulants such as heparin. And you can see in panel B, once a mast cell has been activated, it releases all of these little granules in a very explosive reaction that happens just within seconds. And you can see the granules remain slightly insoluble. They're, on the surface of the slide here, but you can imagine in the body these really act like a slow release pill where the molecules that are inside the granules very slowly diffuse away and have activity inside the tissue that is pro-inflammatory. So at the bottom, you can also see how mast cells respond to dengue virus. On the left, again, the control tissue that's stained with a probe for the mast cell granules. And you can see that the granules are very tightly packed around the nucleus of each of these two mast cells in the control tissue. But on the right-hand side, the mast cell that is activated by dengue, and you can see that the dengue antigen is stained here in red using this antibody 4G2, which is a dengue-specific, envelope-specific antibody. We can see that all these little tiny granules have been released in the surrounding tissue. So I'm going to, for the sake of time, just summarize some of our early findings on how mast cells respond to dengue. And that is that we saw that they're very highly protective in the skin during cutaneous dengue infection. So when the mosquito injects, mast cells are activated, they release these granules. These have a pro-inflammatory effect in the skin at the local site, and that promotes viral clearance at the early stages. Particularly, we get recruitment of these cells in the corner, NK cells and T cells and NKT cells, which clear the infection and prevent the virus from spreading to the draining lymph nodes to the same extent. So we get much lower levels of early viral infection in animals that have mast cells compared to mast cell deficient animals. And many of the mediators that are involved in the recruitment of these cells we highlighted here on the side. And these are also de novo synthesized in addition to the granules that mast cells have with proteases inside. So, but today I want to talk about the pathology that's associated with dengue hemorrhagic fever. And when I first started this project now, more than 10 years ago, we were questioning, this unique spectrum of disease you see with dengue actually has some similarities to other pathological contexts, particularly allergic diseases and anaphylaxis. So you can see here on the left in panels A and B how this individual who has dengue is experiencing a pretty severe rash on their skin. And in the panel below that, how this patient has bleeding into the eyes. So this is microvascular hemorrhaging here in the eyes. And these sorts of things happen as well during very severe allergic reactions. And in those conditions, mast cells, we know, are very essential for disease, as I mentioned, in part because they release all of these mediators, including things such as histamine, TNF, the proteases I talked about, leukotrienes, which are also important for vascular effects, heparin, again, an anticoagulant, and others. And we know that together, these mediators can break tight junctions between endothelial cells. They can reduce clotting. So based on that, we wanted to first question whether mast cells are contributing to vascular leakage during dengue infection. And for this, we use mice that are deficient in mast cells. They have a genetic deficiency, so they don't have any mast cells. And in this case, we saw that when we measure vascular leakage, here in this particular panel on the top right, we're measuring vascular leakage by hematocrit, which is the concentration of the blood that is red blood cells. So you can imagine that as you lose the fluid in the blood vessels, as the plasma seeps out through blood vessels that are more permeable, you end up with a higher concentration of red blood cells that are remaining in the blood. And so this hemoconcentration is observed in wild-type mice that are dengue infected, but we do not see this in mast cell deficient mice. And this strongly points to mast cells as being an essential contributor to vascular leakage in this animal model. And down below, we also wanted to look in humans and try to determine whether mast cells are activated in humans. For this, we had a group of patients in Singapore, and very uniquely, these patients could be classified into having dengue fever or mild disease, or dengue hemorrhagic fever, which is really severe disease. And initially, we wanted to just use a biomarker of mast cell activation. And this, we particularly used this protease, which is called Chymase. This is a really mast cell specific molecule that is made by mast cells and stored in their granules and not made by other cells. And we thought it can be a biomarker of mast cell activation, and we'll understand then if humans are experiencing mast cell activation during dengue infection. And so indeed you can see both patients with DHF and dengue fever as well have heightened levels of mast cell activation, but one important observation was that this seemed to correlate with severity, so that those patients with dengue hemorrhagic fever had much higher levels of Chymase in their serum than patients with a milder dengue fever. And this really suggests that mast cell activation itself is correlated with the severity of disease. So one other thing I told you at the beginning is associated with severe disease is previous infection. And there are many different ways that it is thought that antibodies could possibly contribute to severe disease when you have a secondary heterologous infection. So this cartoon shows this. You can see during a primary infection, infection by one serotype will generate an immune memory. And part of that memory response includes the production of antibodies that are specific against the first serotype. And again, on the right-hand side at the top where it says secondary homologous infection, what this diagram is showing is that during a secondary infection with the same serotype, we are able to see neutralization and blocking of infection by these antibodies. So they're very effective in preventing reinfection. But during a secondary heterologous infection, it's a very different story. First, we can see a phenomenon that's called antibody-dependent enhancement of infection. And in this scenario, we know that when you have infection with a second serotype, you can get binding of these antibodies to that serotype, but they may not be protective. And in fact, they can cause a process called opsonization, which is when these immune complexes of antibody and virus are taken up by cells that express the FC receptors, such as dendritic cells, monocytes, or macrophages. And this can actually lead to enhanced infection of those cells, leading to worsened severity of disease. These antibodies can also, over here on the far right side, you can see decorate infected cells and lead to NK cell mediated killing of those cells, which could induce a great deal of cytokine release and inflammation associated with the death of these infected cells. But what I'm going to talk to you about at length today is this process that is depicted right in the middle at the bottom of this slide, which is mast cell mediated activation by immune complexes. And we're going to talk about how we show that this occurs in detail. So we know that mast cell activation is enhanced by IgE, but an important point is that it's also enhanced by IgG. And so you can see in the top graph where we have used increasing concentrations of this antibody 4G2, which again I mentioned is a cross-reactive antibody that binds to all of the different serotypes of dengue. You can see that in the presence of increasing concentrations of antibody, we have increasing mast cell degranulation. And that's shown here at the bottom as well, where in the control cells, the granules are very nice and tightly packed inside the mast cells. But with dengue activation, we begin to see release of these granules onto the slide around this mast cell that's been activated by the virus. And then on the far right, you can appreciate that this is much more extreme in the presence of a enhancing antibody. Well, it's not only the mast cell degranulation that's enhanced, it's also the vascular leakage. So when we use this in vivo model, SASH mice here are to indicate the mast cell deficient mice, and we compared them to wild type mice, and we measured their vascular leakage after dengue infection. So on the left, we use hematocrit, which I spoke about previously, and on the right, we used Evans blue dye, which is injectable dye that you can then detect as it leaks into tissues. And again, in the wild type, which is shown here in magenta, we can see that there is a significant increase with dengue alone when we give it with just an isotype control antibody that's not specific, and that we saw previously as well. But in the presence of antibody, we get greatly enhanced vascular leakage. And again, this is shown by both of the methods. In contrast, in the SASH mice, which lack mast cells, not only do we lose the direct vascular leakage that's mast cell dependent, so that the infection alone induces vascular leakage that's really close to the baseline, but again, we lose the antibody-enhanced component of that as well. And again, that's shown with the turquoise color in both of these two scenarios. So an important point is that this is really happening through the mast cells receptor for IgG. So you can see on the left-hand side, we treated mast cells with this 4G2 antibody that cross-reacts between different dengue serotypes, and we challenged them with the mast cells then with dengue. And you can see that both dengue alone and the UV inactivated dengue induced a significant increase in degranulation above the baseline level caused by dengue alone. And then, the overall baseline is shown by this gray line at the middle. But when we used mast cells, particularly bone marrow-derived mast cells that lack the Fc gamma R3 receptor, which is the activating Fc receptor on mast cells for detecting IgG immune complexes, we can see that this antibody-dependent enhancement is lost. So again, we can test this in vivo. So we made mast cells from either wild-type mice or Fc gamma R3 knockout mice, and after generating those mast cells, we injected them into mast cell deficient mice. And this allows engraftment of those mast cells. And in that context, we can then see the function of the mast cells in vivo in animals themselves after infection. We also then sensitize these animals with an antibody against dengue, followed by a dengue challenge. And so you can see here in those animals that are reconstituted with the Fc gamma R3 knockout bone marrow-derived mast cells, we see far reduced vascular leakage, again, in terms of the hematocrit readings as well as the dye leakage into the tissues. So this really suggests that mast cells can be activated by immune complexes, that antibodies are able to enhance the mast cell's effect on the vasculature during dengue infection. And then we also went back to our dengue cohort and we looked at those patients that had DHF. And we split those patients into those that were having a primary dengue infection and those that were having a secondary dengue infection. And this was determined serologically in advance. And we saw that when we compare the levels of Chymase, which are analogous to the levels of mast cell activation, it's much heightened in the context of secondary dengue infection compared to a primary dengue infection. So even for those severe patients, the ones that are having a secondary infection are having much higher levels of mast cell activation. So just to summarize this part so far, we can see that mast cells mediate enhanced pathology during secondary dengue infection, that dengue-specific IgG can augment the release of mast cell products. This seems to happen through activating Fc receptors on the surface of mast cells. And we think this is another mechanism for how how antibodies can exacerbate immune pathology during secondary dengue infection, and one that is mast cell dependent. So I still want to emphasize that early in infection, we do know that mast cells are highly protective when the infection is localized and it's early in the disease time course. But we also see that their products can contribute to severe disease. And I think this is very similar to many other immunological contexts that we think about, where we know that the immune system is protective. It does protect us against infection, but that occasionally when it's activated in a heightened way or an aberrant way, it can lead to immune pathologies that are detrimental. And so the next question we had was, which of these products that are elicited by dengue from mast cells actually are causing this vascular leakage and hemorrhaging? In particular, we focused in immediately on something that is mast cell specific. We wanted to look at the mast cell proteases because we thought, while many cytokines and lipid mediators can be made in the context of infection by numerous cell types, if we're looking at something mast cell dependent, we really wanted to view it from the perspective of understanding which mast cell specific products could be contributing. So two of the products that we focused on are proteases, tryptase, for example, and chymase. Chymase is an angiotensin-converting enzyme. Tryptase is known to cleave PAR receptors between endothelial cells, and both of these have other functions in vivo that are pro-inflammatory. So to begin to understand which of these could be inducing the endothelial permeability that we observed during dengue, we started by culturing primary endothelial cells as a monolayer. And you can see we imaged them here. We stained them for the tubulin, which allows us to see their structure, the DAPI for the nucleus, and also ZO1, which is a tight junction protein that you can see just is found at the border of the cells. It's important for holding these cells together. And when we treat the cells that we've created as a monolayer with either tryptase or chymase, we observed that both of them were able to induce some degree of permeability of these monolayers. So, for example, on the left-hand side at high concentration, you can see that tryptase has really completely eliminated this ZO1 staining at the border of cells, and we can even see cells peeling up off of the tissue culture plate. In the lower concentration, we start seeing some punctate staining between cells that really is indicative that these tight junctions are beginning to be broken down, and that's similar to what we see at high concentrations of chymase. We also were curious if this happens in vivo. So to begin with, we produced tryptase and chymase as recombinant proteins to inject into recipient animals in the absence of dengue infection. And what we observed is that given the same concentration of tryptase or chymase, there was a significant increase in the hematocrit or vascular leakage in vivo that was entirely dependent on the protein itself. Importantly, this tryptase and chymase were given at concentrations very similar to what's observed in DHF patients, and we saw that at the same dose, tryptase was much more effective in inducing vascular leakage compared to chymase. And this did not occur with an innocuous control protein, which we used here, which is ovalbumin. Similarly, we also borrowed from some of the allergy and anaphylaxis literature, where a drop in temperature is a key marker of shock and can be used to read out the presence of shock after anaphylaxis. And so we wanted to use that system here. When we injected tryptase and chymase, we noted that there was a very significant drop in temperature that's indicative of shock that occurred at the same level of tryptase that is observed in DHF patients. So this really showed us that both tryptase and chymase can induce vascular leakage, but tryptase is much more strongly associated with shock. And this also occurred in humans, so we used some serum samples to test for tryptase. And these patients were graded according to the severity of the DHF that they experienced. So grade 2 DHF is associated with vascular leakage and hemorrhaging, but not necessarily with shock. Grade 3 DHF is associated with shock, but grade 4 DHF is very severe. It involves shock in the absence of a pulse and is indicative of circulatory failure. In contrast to that, fatal DHF is really very similar to grade 4, and there's not a distinguishing feature other than the survival. So you can see here that there's a very strong correlation between the levels of serum tryptase and the grade of shock that the patients were experiencing. So then the next question is, of course, if mast cells are contributing to dengue immune pathology, are we able to target mast cells to limit the disease? And this is an exciting possibility because we know that there are many drugs that are on the market already that target either mast cells or their products, and they've been used in the clinic for decades, and they're very safe. So some of these include mast cell stabilizers, such as chromalin and ketotifen, leukotriene receptor antagonists, and TNF blockers. And so we wanted to test different types of drugs and identify those that might be effective against dengue. So this is one of our first studies we did. You can see that in animal models that were infected with dengue, we treated, this is a mouse model, with either chromalin, ketotifen, or TNF. We also used separately, that's not shown here, the leukotriene receptor antagonist. And we saw that only the chromalin and ketotifen were very highly effective in blocking vascular leakage by both of our different methods in terms of Evans blue dye leakage into the tissues as well as the hematocrit. We also know that in animals that are infected with dengue, we're able to detect very striking changes that occur with respect to the transcriptional processes happening in the spleen, shown here as an example, but also the liver and other target organs. And many of these pathways that are induced, such as the interferon response pathway, cytokine responses, and lipid metabolism, we saw were reduced or reversed in the presence of ketotifen as a mast cell stabilizing drug. We also used a tryptase inhibitor. We used a drug called nifamostat mesylate, and in this case, we gave the animals nifamostat mesylate and tested the enzymatic activity of tryptase. We saw in panel B here that it was reduced, and when they were treated, we could again measure the vascular leakage, and you can see in panel A a very significant drop in vascular leakage in the presence of nifamostat mesylate. And this really affected the vascular leakage specifically. For example, another sign of severe dengue is thrombocytopenia, or a drop in platelets, and you can see that was not affected. So this effect on tryptase was very specifically associated with the preventing vascular leakage. And in panel D, you can see that this inhibition occurred without greatly affecting the viral titers itself. And what this looks like in vivo, we can measure with a video the vascular leakage over time. So over time, you can see in the control in the very top panel, we just see the blood flowing through the blood vessels, but we don't see vascular leakage at the steady state. In the presence of dengue, however, over time after dye injection, you can see that these images captured at different times show the gradual leakage of the dye into the tissue itself, but this is completely prevented in the context of nifamostat mesylate treatment. And that's just shown here quantitatively, where over time you can see the green line shows an increase in the leakage of the dye, whereas the blue line shows that this nifamostat mesylate is able to completely protect against vascular leakage. So some key points I wanted to make to summarize are that mast cell proteases are really key for dengue-induced vascular leakage. We know that the mast cell activation is a strong correlate of dengue severity. One point I'd like to make is that most of these serum samples that we collected and that I showed you, that data was collected at very early time points, so before the patients actually developed severe disease. And so we think Chymase is a really strong candidate as an early biomarker of disease severity, and that perhaps some of the processes that are being initiated by inflammation early on are Chymase dependent. And then again, we see tryptase, which is not as strong of a biomarker just because of its kinetics of breakdown, quick breakdown in vivo, but it seems to be very important for actually developing vascular leakage, and is strongly associated with shock particularly. So between these two proteases, we think that these are possible promising therapeutic targets for preventing severe dengue, and this is something that our group is continuing to study. So I just want to summarize a bit what we know about vascular leakage during dengue and the immune pathology associated with it. So we know that there are innate processes that can regulate vascular leakage. For example, when the virus infects target cells, we know that we can have the production of pro-inflammatory mediators such as interferons, such as cytokines, and the virus proteins themselves, including products such as NS1, they may directly act on various cell types in the body and cause some aspects of the pathology that we observe and associate with dengue. We also know that people who are having a secondary dengue infection can have more severe disease. And I talked about several ways that antibodies can do this, both by enhancing infection and also through enhancing mast cell activation. But T cells are an important component as well. Perhaps the T cell response and the memory T cell response can also lead to enhanced activation and release of pro-inflammatory mediators in the context of secondary infection. And some of these could contribute as well to the pathology. But I think central to what we have studied is the mast cell, which really bridges this innate and adaptive response, being able to both detect dengue at baseline as well as respond to it in an enhanced fashion in the presence of antibodies. And that these products that are released, mast cell proteases, which are pre-stored and may not be detected as part of a transcriptional response that is measured, are also contributing factors to the vascular leakage that we observe. And so with that, I would like to acknowledge my lab, particularly Chymase and Wilfried, who did much of this work, Abe Rator, who's now at Duke University, did all of the tryptase work, and Juliet Morrison, who is now at UC Riverside, has been very instrumental in helping us look at our transcriptional responses. So with that, I will finish. Thank you.